So this is Dr. Poe, and I wanted to introduce you to the concepts that we're covering in microbiology this week. And of course, these are going to be starting with, of course, what is microbiology and why do we need it? History of microbiology, very briefly, cell types and biomolecules. I'm not really going to cover biomolecules because we've covered that in chemistry, which is a prerequisite for this course. And also the 5 I's. So this is just to give you an idea, and this is to supplement the reading. I really recommend that you read the provided lessons on Canvas, because they have these interactive components that's really going to make these concepts come alive to you. Okay, so let's go ahead and jump in and get started. What is microbiology? Microbiology is the study of microscopic things. And so these are really not visible to the naked eye. And so they're going to be on itty-bitty scale. So we know from chemistry, we have scales like kilo, hecto, deca, the base unit, deci, centi, milli. Well, we can still see like a millimeter, especially in talking about length. So in microbiology, 3 spots down from that, so 1,000 times smaller than milli is micro, which in science is written as this mu. In nursing is little mc, like a microgram would be little mcg, or a micrometer would be mcm, micrometer. So we're talking for length, so this is micrometer scale. And then 1,000 times smaller than micro is nano, and 1,000 times smaller than nano is angstrom. So this angstroms is really the size of bonds. And I think I might be missing pico. But anyway. So microscopy and microbiology kind of looks at these things that are so small that you definitely need a microscope in order to view. Okay, and so this leads us into the history of microbiology. The first person to really use the microscope was this kind of like Dutch guy, Leeuwenhoek. Antony Leeuwenhoek, which I'm totally butchering his name, but that's totally fine. He was the first person to look at, to use microscopes to view just whatever. So he was looking at slices of cork, he was looking at his particular manly bodily fluids, which is disgusting to think about, and then he was also looking at water sources, and that what he saw were these animalcules, is what he called them, absolutely itty-bitty things. We didn't have a term for it at the time. Hooke coined the term cell later, but he called them animalcules. But really, we know them today as these are our microscopic organisms. So he was able to see a large number of them. What he was not able to see were viruses. So viruses is not on this list because you need an electron microscope to see viruses because they are so small. They're over in this range, viruses. He was able to see things more in this range. So he was able to see, of course, bacteria, which are single-celled, no nucleus. Some of them cause disease, some of them are found on our skin, and in our gut and are beneficial. So we saw a lot of those. He could see some microscopic fungi like yeast. Definitely could see yeast. Yeasts and molds. What else could he see? He could see protozoa. Some of them protozoa. Some of them can cause diseases, but a large number of them don't. What else did he see? He saw algae. Algae are plants. He also saw maybe some archaea. So archaea are also single-celled, no nucleus microorganisms that we call prokaryotes, which includes the bacteria and the archaea. Those are our prokaryotes. And then these guys are our eukaryotes. So you are a eukaryote. They have nucleus and organelles. They are large. And there's several other differences as well. The prokaryotes have no nucleus, so it's kind of weird to kind of define something based on something it doesn't have. But nonetheless, here we are. So prokaryotes have no nucleus, and these are typically small. And they all have cell walls. And they're almost always unicellular. I actually can't think of an example that's multicellular. Whereas eukaryotes, I didn't write anything about cell walls because some of them have cell walls and some of them don't. I didn't write anything about unicellular or multicellular because you can see both. The fungi can be unicellular or multicellular. The protozoa are typically unicellular. Algae are typically multicellular. You are multicellular and you have a nucleus, but you do not have a cell wall. Plants have cell walls. And we're all in the same category. Okay. So that's Leeuwenhoek's contribution. There's also several other historical contributors to my microbiology, and I'm just going to mention one more, Carl Woese, and I'm mentioning him because he's important in differentiating actually different groups of prokaryotes. There's another Carolus Linnaeus, who helped make the taxonomy and how we're naming these different organisms. And we end up with this binomial nomenclature, which you've seen before, where there's like a genus name and a species name, and that's the scientific name, like Escherichia coli, Homo sapiens. Those are species names. Well, anyway, so that's the other Carl. This Carl Woese, he was really looking at things like Escherichia coli and Methanobacter archaea, and also the eukaryotes. And he wanted to find a way, like, let's kind of like make this new tool. And what he did is he looked at ribosomal RNA sequences, and he looked at those because every single cell that we know of has ribosomes. Remember that ribosomes are important for making proteins. You absolutely have to know that. Ribosomes make proteins. If you're a cell, you have a cell membrane. You have, so I'm going to draw that out for you. So every single cell has a cell membrane. Cell wall is optional. It has some cytoplasm, the fluid on the inside. Cytoplasm, cell membrane made of lipids. It's a phospholipid. And then it's got some genetic material. That's our nucleic acids. And then it's got some, replication machinery that can copy that for transcription and replication, and also ribosomes for translation. Okay, so when we're looking at ribosomal RNA, these make up the ribosomes. And those ribosomes give us proteins. Okay, so we absolutely have to have those in cells. This is a great thing to look at because it's got, these are encoded in the DNA, and every cell has them, so it's a really great way to kind of compare these different things. And so when he compared it, what we kind of expected was to end up with a dichotomy of like prokaryotes are going to be over here, and eukaryotes are going to be over here and be completely distinct. But what he ended up seeing was this. And you think that the two prokaryotes would be together, but that's not what we saw at all. The eukaryotes are here, that's us. Some of them have cell walls, some of them don't. And it turns out the archaea, which were a type of prokaryote that were always looped in with the bacteria, they're more closely related to the eukaryotes than to the bacteria. So this is like our family tree, and this is our last universal common ancestor. This was like the very, very, very first cell. Somewhere back in history, there was a branch, and these guys to the right got bigger because we know eukaryotes are big. And these guys stayed small. And they have one cell, so they're single-celled. So that happened somewhere back in time. But these guys are also small and single-celled. So they looked like the bacteria, but it turns out genetically they had diverged. This is what this breaking point is. There's a divergence. An event happened here that these guys stayed small and single-celled and looked like that, and these guys are a different kind of small and single-celled. And we see that in the ribosomes. The ribosomes here for the archaea are more similar to the eukaryotes than to anything else. Okay, so then there was another evolution point here, and then the archaea branched here, and these guys became like multicellular and big, and archaea stayed small and single-celled. And so when we looked at these molecular differences, it became more apparent. Bacteria cell walls are made of one compound called peptidoglycan. Archaea is made of a different compound, pseudopeptidoglycan. And then eukaryotes have like completely different chemical structures. So that's just Carl Woese. So I just talked about Leeuwenhoek and Carl Woese. Okay, so covered microbiology, history, cell types, and biomolecules as we were talking about it. Okay, so there's a lot more detail, of course, into the individual structures of bacteria, archaea, and eukaryotes, which we're going to talk about in class. So what I wanted also to mention briefly in this video, because this is a little bit confusing, bacteria are typically found everywhere. There is not an environment where we haven't found bacteria. Archaea typically survive well in extreme environments. Because of that, they are typically not involved in disease. If you think about where these can survive and grow, our body is not an extreme environment. Our pH is close to neutral. Our body temperature is not that high. That we're not an extreme environment. So if these live in extreme environments and thrive there, they're not going to live in our body. So therefore, they can't cause disease. Eukaryotes, these are also found in many environments, and these are also extremely diverse environments. But typically not that extreme. So we find these in like oceans and some in your guts and fresh water and salt water and soil and those types of things, but not so much like volcanoes. Volcanoes is more like archaea, bacteria, that sort of thing. Also, I wanted to mention that there's, so a lot of people feel like, oh, bacteria, that's really, they're focused on disease. We only know of about 70 species cause disease. That's like 1%. I'm totally making up that number, but it's a very small number. So most of the bacteria that you come in contact with are actually not going to cause disease. And these typically don't cause disease. And some of these do cause disease. So obviously the focus for this class are going to be on the differences in these three groups. And eukaryotes are very diverse, and we focus a lot on theirs. Archaea we don't know a lot about. Because they don't cause disease, they're not studied as much. Bacteria we know a lot about, so we're going to focus on those a lot. Okay, so back up where we're at. So in microbiology, we are going to be using the 5 I's to study microorganisms. I also want to mention organisms that one of the groups that we study that we're going to cover in this class are the helminths. They're worms. They do have a microscopic lifestyle, and we also talk about mosquitoes a little bit because they carry like Plasmodium parasite that causes malaria. So we do talk about a couple other like large eukaryotes. So they wouldn't be applicable to the 5 I's, but everything else will. So our 5 I's. Inoculation, that is taking a microorganism from a sample, whatever the sample is, and putting it into a new sample. So that could be a new broth, that could be a petri dish, however we're doing that. Once we have inoculated it, and we typically use a loop, and we're actually doing that this week in lab, to transfer some bacteria from here to another type of culture, then we incubate it. So the next step is incubation. So what that does is it takes whatever we've inoculated and we grow a whole bunch of those cells. Because these are so small, we can't study individual cells. We need a whole bunch of it. So these liquid broths are really great for growing a whole bunch and then doing other things with. And these plates are really great for growing individual colonies because plates are solid. They give a solid surface for the growth of colonies, and the colonies are different for each individual bacterium. So some of them could be red, some of them could be green, and this is how we can study them. So they will look different, and yes, we are practicing this in lab. Okay. Okay, so that's the point of incubation. Then from there, we do isolation. So if we have a whole bunch of things growing in a broth, we can't tell what's growing there. What is in here? Or if we have a whole bunch of things growing really densely on a plate, we can't tell what's here. We need to get individual colonies. So we isolate the bacterium. And so we can do that using sterile technique and loops. And we can do this with the spread plate technique, which makes dilutions. Basically, the goal here is to make dilutions and to view individual colonies, because when we get to individual colonies, they are completely isolated. So there's several techniques mentioned in the book. There's the spread plate technique, which we are not going to do. There's the pour plate technique, which we are also not going to do. My favorite is the streak plate. Streak plate, where you take, and we're going to be doing this in lab, and I made a separate video of it, and it's basically where you take quadrants. It's also called the quadrant method, and you streak it out a little bit, and then you use those to make the next quadrant, and then you use those to originate the next quadrant, and then finally the final quadrant. And then the hope is that at the end you will end up with individual colonies so that we can look at them and tell what it is. So this has to be done on plates because that's the solid surface. After we have isolated our samples, then we can do our inspection and view them. Inspection. And we look at the individual colonies. And then we can also, and that can give us some idea. And then we can also use microscopes to look at individual cells, which is a very specialized tool, very expensive tool. And so we're going to start doing these in the Unit 2 labs, and there's the different, and then when you're looking at things in the microscope, you want to look at it under different staining techniques because it makes it easier to view. And we can also do different types of staining, tells us different features. So simple stain is just going to show us, that's just like, it's fast and it doesn't show us anything other than like maybe cell shape and cell arrangement. So cell shape is going to be like rods, spiral, sphere. And then cell arrangement is how they are attached. So are those rods attached together like that? Are the spheres clustered together? So so arrangement means how cells are attached. Are they in chains? That's called strepto. Are they in grape-like clusters? That seems pretty random. That's called Staphylo, and this one's called Strepto. This one, of course, is also a long chain arrangement that would also be Strepto, and it could be tetrads, which are groups of 4. There's others also listed in the book. So arrangement means how those are arranged, and this is, of course, the product of cell division. This is how those cells are attached to each other. So those cells not fully attaching at the end of cell division gives them these different specific arrangements. We've also got differential stains. Differential stains, such as the Gram stain that we're going to perform in lab, that tells us information about if it's Gram positive or Gram negative, and this relates to the amount of peptidoglycan in the cell wall. So, of course, this would not be applicable to eukaryotes, which absolutely do not have peptidoglycan in their cell wall. We can also do an endospore stain, which tells us whether or not they have endospores. Endospores are very difficult to get rid of. Things like anthrax, Bacillus anthracis have endospores, and these can only be destroyed by autoclaving. So that's important to know if they have that. So the 2 groups that typically have that are Bacillus. What's the other one? Clostridium. So if you've heard of C. diff, lots of those have endospores. And then finally, last group that we can look at are the acid-fast type of cells. And so the types of cells that have these, they have mycolic acid instead of peptidoglycan in their cell walls. These grow slow because that acid is hard to make. These are found a lot in Hansen's disease, the mycobacterium, because you see the myco for mycolic acid. These are their microbacterium that cause leprosy, also known as Hansen's disease, and they also cause tuberculosis. So those are important things to know, and we can tell this from the different stains. So these differential stains, these are typically 2 stains, and the different colors that they turn tell us these different things, and we'll talk more about that in class. There's also different types of microscopes, and there's more different types of staining as well. There's also negative staining. I want to mention that. Negative staining tells us about capsules. Anything that has a capsule is absolutely this is a way to avoid phagocytosis. This is a virulence factor. That's a V. Virulence factor, which means it causes disease. Okay, and then there's other stains as well, which we'll talk more about in class. Okay, there's also several types of microscopes. Which your book does a wonderful job of, and we'll discuss more in class. And so the questions that you want to ask, is staining required for the type of microscope? If staining is required, that's 100% going to kill your sample. Can it view live samples? Sometimes it's important to look at something that's alive if we want to see how it moves and how it infects and things like that. How small can it see? I mentioned earlier that electron microscopy is good for viruses, and we could not see viruses with light microscopy because the resolving power wasn't good enough. So there's the book has many examples of that, which we don't necessarily have to go to. Okay, so that's all inspection. And so we use all of these steps to get to identification. That's really the goal. And the final identification step, we can also supplement that with like our biochemistry stuff like DNA sequencing. That's definitive to tell different species sequences. We can do biochemical tests, some of which we will do in class, like fermentation tests, resistance to antibiotic resistance. And there's several others too. Okay, so those are our 5 I's to get to the final goal of what microbe is this? How does it live and what does it do? Okay, so I hope that this was an okay starter. Be sure to go back and read through the lesson, read through the textbook, and review the questions at the back of the textbook for additional kind of like quizzing yourself, additional study types of things. Thank you so much for listening.